years, and now we want to highlight this topic with 3 presentations, and also we'll have quite some time for questions afterwards. So just 2 things to start with. On a formal side, please switch your micro off if you haven't done so. We will record that meeting, so it's also available for those who couldn't attend today. If you don't want to be recorded, switch off your camera and rename yourself. We'll try to do that in presenter mode, so it's only the host who's going to be presented, so not everyone listening, but in case you have any strong reasons not to do so, we want to say that before. First, I would like to hand over to Cristiano Vernisi, the chair of the cost action, to briefly introduce you to the G-BiKE project, and then we'll dive right in the middle of the 3 main talks. Cristiano, please.
Okay, good afternoon, everyone. Thanks to Gernot, Elena, and Pamela for organizing this very interesting webinar. And so my duty today is pretty simple, just giving you a brief overview of what G-BiKE is. So first we start by the name. It's of Genomic Biodiversity Knowledge for Resilient Ecosystems. G-BiKE is a cost action, so run under the funding of the European Commission. Okay, so we are basically in this basic layer of the biodiversity as defined originally back in 1992 at the Convention on Biological Diversity in Rio de Janeiro. And what the main aim of the G-BiKE? So basically, we thought to establish the use of genomic data and genetic data as a standard routine tool for monitoring and managing a wild and exotic population of plants and animals. And of course, we would like to put the genetic diversity component in a central place in many relevant European policies, such as the European Biodiversity Strategy 2030, the management of Natura 2000 sites, and the Habitat Directive. How to do that? Basically, thanks to previous experience in which many of us were involved, we understood that the situation was like having two separate wheels. On one side, we had the practitioners. On the other side, we had the scientists. Too often, the two wheels were just disconnected, so not making any opportunity for movement. So we thought that maybe G-BiKE could offer the opportunity, for reason or using the bike metaphor, the opportunity to put together the two wheels in a proper bike, so putting the frame to this bike, and maybe why not trying to move forward. As usual, also our project is organized in different working groups. You can see here the name of people being in charge of this. Unfortunately, you can see also here the name of Mike Proofer, who recently passed away, to whom most of our last activities will be dedicated. The basic tools offered by cost action are for networking. So no funding for basic research, but workshops, training schools, short-term scientific mission, virtual mobility grants, webinar conferences. We managed actually to have the participation of all the 40 countries being involved in the cost association, which I think is pretty outstanding, and also demonstrating the interest of these topics. And I will say at the very beginning, the direction of this kind of journey with the bike is really to arrive in front of the G environment and the G research in order to convince them to put the genetic diversity in the central place, the deserved place for any action, any policy related to biodiversity preservation, safeguarding, and why not also the sustainable exploitation. But of course, while running G-BiKE, we were aware about something that was going on a larger scale, which is exactly the scale at which the Convention on Biological Diversity engages itself. And so we thought that it would be the right opportunity to look not just on a continental European scale, but broader. And that's the reason why we made a lot of effort towards the effective implementation of the genetic diversity concept into the new, the so-called post-2020 global biodiversity framework. Of course, I need to make a long story very short here. I just can tell you that apparently, not only apparently, but also officially, we managed to have finally genetic diversity acknowledge the role it deserves. So now we have genetic diversity fully and explicitly mentioned in Goal A of the so-called Kunming-Montreal Global Biodiversity Framework ratified on December 19, 2020. Second, then we have the target 4, which is about also the maintenance of the storing of genetic diversity within and between population of all species, which I think is very relevant. So insofar, in the previous framework, the mention to genetic diversity was only about agricultural or farming relevant species. And last but not least, probably one of the most important achievements is that we have a specific and exclusive indicator for genetic diversity, which is the proportion of population with a species within a species with an effective population size above 500, which is a kind of threshold we selected according to several previous scientific evidences. Okay, of course, this is just the beginning, I would say, even though the G-BiKE action is going to end this September, but together with the IUCN Conservation Genetic Specialist Group, the Jobon Genetic Conservation Working Group, and the Society for Conservation Biology, 2 years ago, we created the Coalition for Conservation Genetics. We're going to apply for new funding, and hopefully we are also trying to maintain the G-BiKE not only logo, but G-BiKE spirit in sense, the network by establishing after the end of cost action a joint research unit. And so this is also an invitation to all of you, if you are interested in taking notes about this and to address to me or to Gernot, to Elena, or other G-BiKers about this possibility, because actually we really think that it is now the time that we are in the process of implementation of the global biodiversity framework. So we really need to give our expertise, our help, and what we know to people, but are requested to respect the CBD regulation. And after saying this, I would like to thank all of you, and I'm really keen on listening about the next talk. Thank you very much.
Thanks, Cristiano, for giving the background information on G-BiKE and setting the frame of this talks. A very special welcome to all of you being here now and interested in topic and listening to the following 3 talks, which will be focused on kind of 3 main topics people have been interested in, how genomic and biotechnical tools could be used for conservation. It's my pleasure as the first speaker to invite Maud Quinzin. I hope I pronounced that right, from Belgium. She's been involved in very different projects and will give a first introduction. Please, Maud, present, share your screen, and then I'll hand it over directly to you. One formal thing, you can already start typing questions in the chat. We will summarize them and answer them in the end, but you can also kind of write them down. We will also have a Q&A sessions after these talks. We will run each talk, and after all 3 talks, we'll have a joint Q&A session, but you can already start typing potential question in the chat. Please, Maud.
Hi, everyone. Thanks, Gernot, for the introduction, and thank you, everyone, for joining. So I have a very hard task of talking about a few genomic advances and gene editing for conservation in 15 minutes, which is impossible. So everything is going to be very light, not in detail, but feel free to ask questions if you have, if you want more details on something. But yeah, it's not going to be very exhaustive. So I guess I need to start with the advances in genomics and why it is important now to know about those in conservation, because of course these tools are becoming more democratic and accessible. So more and more genomes are available for more and more different species. Also, there is an improved analytic power. So for example, we have more access to different kinds of DNA, including degraded DNA, which is very useful when you work with museum samples, with samples that come from trafficking places, or just to monitor populations when you don't want to have a non-invasive sampling. So for example, collecting hairs or scats from bears, wolves, anything. And then so more genomes available, more and more, and also an advantage when you want to compare data among labs. That said, that's a lot of new data type. That's a lot of information with a lot of good quality information, but it is still adding a complexity. So now it is even more complex to analyze those data, and it remains a costly adventure to go into genomics. You need an expert to analyze those data and to actually do the DNA preparations and everything. So the gap between the researchers, scientists, and the practitioners on the ground is definitely it can be increased. And so that's why a talk like this and a presentation, those presentations are really important, so that we make sure that we understand where is the gap and what has to be explained in more detail to make it useful. So opportunities with genomics. This is really not an exhaustive list. It's just some things that we as researchers are starting to see evolve. And so, for example, when you have a population that is endangered or others, what is really important is that you can now detect adaptive parts of the genome and screen for individuals who are more adapted to the conditions we are looking for or that the ecosystem is experiencing. We are also more likely to detect hybrids and determine if they have good impact. And so if there can be an opportunity for genetic rescue, for example, or if they have a bad impact and then they should be removed from the environment. Of course, one of the main and most easy part of understanding where genomics can help is improving the accuracy of all the diversity estimates and demographic parameters that are relevant in our conservation programs. Improving the power with degraded DNA, I talked about it, museum samples and trafficking, monitoring of pathogens and microbiomes are also starting to be very important, and so on. So you have a lot of different things that genomics can bring to the table. And if in this list you find something that is really interesting for you, or that is already challenging for you, please take note. And if you have more ideas, take note, because there will be a poll at the end of the 3 talk where you can actually highlight to us what is challenging to you to understand, or if you need to have a topic where we could help explain more. So keep that in, write it down, and you can mention it during the poll later. Anyway, so okay, genomics in general, bringing a lot of quantity of data and quality as well, but that's not it. The thing is that not only we can characterize diversity much better, we can also have more technologies. I mean, it has allowed to develop technologies where we can alter the DNA of individuals. And 2 very clear examples is cloning and gene editing. Cloning, Sam is going to talk about it in the next talk, so I'm not going to talk about it, but we can talk about gene editing. Gene editing in conservation. So you have 2 ways to go about it. One is to think about providing resistance to a species that is threatened. For example, a species that has a disease and you want to make it resistant to the pressure. One good example is the American chestnuts in the US, where there are variants that are known to be resistant to the or helping to the blight, and so they have been using gene editing to provide those variants to the trees in the US, and are probably going to start using it. And the second way to use gene editing in conservation is by controlling the population of invasive species. And that's where we're going to go next. In particular, the case of rats. So rats are really have been responsible for environmental unraveling across the globe. And for example, on islands, they are the synonym of extinction, and have really decimated population of birds, amphibians, reptiles, plants, native ecosystems in general. And so it's really a huge problem in conservation. And one thing is that the current tools that we have to regulate the population of rats are traps and poison, and sometimes we introduce predators. But the problem with those tools, and don't get me wrong, there have been a lot of successes. And we've seen on many islands that the success of those campaigns of eradication of rats, where the native ecosystem is really returning and healthy now. But the complexity increases with islands being bigger now, and I mean the islands we need to treat are bigger and bigger because we started with the small islands. There is also a problem where there are inhabitants and so on. So there are problems of scale. We can't take helicopters or drones to release poison on entire islands all the time. There is a problem of efficacy, where we know that rats are becoming resistant or they avoid traps and poison. They're really clever. There is a problem with specificity. Sometimes other species are affected by the poison, and so we lose some species due to the poison. It's very costly. A lot of people have to be involved, so it's very hard. And there are a lot of problems with the welfare, because using poison, meaning that we are making those very social animals suffer for several days. And so we have a lot of problems at the ethical level on that part. So that's why we are talking about gene editing, is because the community, the conservation community, is really looking for alternatives that are going to improve those different, or like put aside those different caveats of the management of invasive rats. How would that work? So you have an invasive alien invasive, and the problem that the native animals are endangered, and you use an engineered rodent release. These engineered rodents would spread a negative variant, so it is going to have an impact on the fitness of those invasive animals. And we hope that it can spread in the population, and so that the actual population of your invasive is reducing and the native ecosystem can be restored. So that's the idea. It looks great on paper, but why is it not yet used for rats? Well, there are different things. First, there is definitely a lack of knowledge on the ground. So the thing is that rats are really important. They're affecting food security, by feeding on our crops. There are a problem for health security because they transmit diseases. They're a problem for the environment, for the infrastructure, but we don't know much on them in the wild. So, for example, the cycle, how they react in nature, and that's quite important when you want to introduce a genetic variant, or you want to introduce individuals in a population. You need to understand what is the local, what are the local processes, genetic and at the genetic level, but also at the individuals levels and the population level. So you need a lot of information. But of course, before anything, is that the technology is actually not ready yet. So it is still in development. And that is a good thing in one way, because it gave space for a lot of conversation around different problems to be handled. So there are a lot of problems, ethical problems, political problems, economic problems, social problems, cultural problems, and all these different problems are kind of in the movement now, and they are impacting the design of those technologies. And yeah, so that's very polarizing. A lot of people want to try to use it. Some really don't want to. And one of the main place where there is a conflict is because you have a trade-off to make between invasiveness, because you want your negative trait to actually be pushed into the population so that you can reduce the density of your invasive population. But at the same time, you don't want that negative trait, that fitness negative trait, to be spreading outside of the population you want to regulate. And so that's really what is really a problem at the moment, and one of the main place of discussion on whether we should or not using gene editing. Now, okay, so invasiveness and containment. Invasiveness, we want the trait to, the negative trait to spread into your population. And one of the things that has been proposed for that is using a gene drive. So gene drive, Christy is going to talk about it in the third talk, so I'm not going to go too much into detail, but the difference is really important to understand between gene editing and gene drive. Gene editing is really cutting and rearranging pieces of DNA. A gene drive is the same thing, except that that piece of DNA, its inheritance is going to be altered so that the descendants are going to inherit that variant much more than the regular 50-50 in the mammal inheritance scheme. So it's just, there is a huge probability that if you have different descendants from an individual that has a gene drive and a wild type, or like the descendants of those two, of that reproduction, are going to be bearing of the drive of that rearranged piece of DNA. And so it actually makes that the gene editing part is going to spread really quickly in the population. And that's one of the ways we have to spread the gene editing part and the negative traits in the population. Now, there are ways to make a gene drive more constrained. So for example, we make sure that we edit a part that is really local and that is only found in the population that you want to control. And so if one individual goes out of that population, it may not be, it is not going to affect the other population. There are other ways of doing it, like with thresholds, but I'm not going to go into those details. Feel free to ask questions if you want, or maybe Nikki is going to cover that after. But then anyway, so the design of the technology is very important, and a lot of the discussion is going to be also in terms of the number of individuals that you can introduce in a population. So if you want to spread a negative trait to reduce the population of the invasive species, you have to introduce engineered individuals in the environment. So you actually have to increase for those first moments, for the moments where you have to introduce, you increase the pressure of the predator on the population. And so that's also one thing that is good with the gene drive, is that you would not have to introduce too many individuals. But again, there is a trade-off. And so there are also thoughts of like, do we really need a gene drive to spread a negative trait in the population that we want to regulate? And again, the discussion is around the design of those technologies, and the considerations are really locally treated, ethics, culture, conversation that are still going on right now. So there is an idea of, well, maybe we don't need a gene drive to spread. And an idea here, it's just a simple example to show you how it could work with the gene editing to regulate a population of rats. So here on the left, you have a male that has the gene editing part. He's going to mate with a female that is wild. And the traits that this male is bearing is that the embryos of those matings, the female embryos are not going to develop. So females, they die very, very early on. And the males, though, the embryos, males, they survive. And when they mate themselves with wild females, they also only produce males because the female's embryo die. And so suddenly you only produce males in the population, and that's how you bias the sex of your population so that you only have males in your population. And at some point your population collapse. So that's the idea. And maybe we don't need a gene drive to do it. Maybe we have to. That's again a good conversation to have, but that's a lot of detail. But that's really to give you an idea. And where this is helping is really that you don't have to kill the animals. So you don't have to use poison. You don't have to have these problems of ethics. And so, for example, in Australia, PETA, which is really a big name for animal welfare, they're actually kind of happy that these technologies are seeing the light. And so this is something to consider. Again, it's just because we need an alternative. So considerations to have when you want to think about, oh, should we use gene editing or not? There are problems about the number of individuals that you're going to introduce, because you're going to have a pressure on your population, on the threatened population, on the native species. And so how often are you going to have to introduce them, when and where? These are all questions that you're going to have to answer before really considering doing it. Again, there is the discussion about gene drive versus gene editing and their invasiveness. You have to consider that. Sometimes the technologies that are developed for rats or for other mammals could be used for other mammals. So if you think about mammals, like if you have a problem with mammals, consider it and talk to those specialists working with rats, and because maybe there is a solution. It is going to be different every time you change the sex determination. So birds are going to be different, insects are different, hymenopters like wasps are going to be different to treat, because the transmission of your negative trait is going to work differently. But maybe you're going to have more of that in the last talk. And then one thing is that really important to understand is that gene editing is definitely not a silver bullet. It's part of a more integrated strategy. So for the moment, people are just thinking about it as a complement to poison and traps. And hopefully it can help. It can be making it efficient, but we're not thinking about it as, again, like as a silver bullet. Technology is not the only solution. And then again, consideration of ethics, culture, politics, everything is really in conversation when you want to introduce that into the conversation. And it doesn't replace, like for genomics. Genomics, you know, it's not going to replace a good study design. It's not going to replace a good biological replicates. So you're going to have to be good at understanding and monitoring your environment. And here for gene editing is going to be pre, during, and post introduction. And then, of course, it represents a lot of work, but we have a lot of background too, like invasive, the introduction of species in an environment to try to regulate species, so biocontrol. We have a lot of background now. We understand more and more those dynamics. And so this is certainly new with gene editing, but there is a lot of lessons learned already. And so if I had to say something with genomics, it's just really, it's a lot of good opportunities, but a lot of work and very interdisciplinary work that is going to be needed to cover those. And I'm going to stop there. Thanks.
Thanks, Maud, for giving that overview on potential of genomic methods. If you have any questions, put them in the chat. We will continue with Sam Risley from the University of Florida and on the famous case study on the black-footed ferret. Please, Sam, share your screen and I hand over.
Well, good afternoon, everybody, and I really appreciate your attention. So black-footed ferrets are an interesting endangered species, almost since they came under the purview of the U.S. Fish and Wildlife Service, which is the governing body for endangered species in the United States. It became a test species for incorporating new biotechnologies into conservation, whether it was reintroducing animals back into the wild, using conservation breeding in a zoo setting, using artificial insemination for reproduction, and now considering using gene editing as a conservation tool. So let me just give you a little bit of history of the black-footed ferret and tell you why it is endangered. So black-footed ferrets are one of 3 ferret species found worldwide. They're the only ferret found in North America. They live in the central grasslands of North America. And those grasslands in the early 20th century were largely converted into agriculture. Black-footed ferrets are very interesting. They're considered an obligate predator of a ground squirrel called prairie dogs. And so prairie dogs were this densely populated species throughout these grasslands of North America that were preyed upon by black-footed ferrets. But not only did black-footed ferrets use them as prey, but they spent 95% of their lives living in these extensive tunnels that prairie dogs made as well. So as agriculture came along, prairie dogs declined, and so did black-footed ferrets. Until 1970s, there were only 2 populations known. And then by the 1980s, there was only 1 population known, which is shown here in the red triangle. In 1982, there were thought to be about 120 black-footed ferrets in this population in Wyoming, but an epizootic of canine distemper swept through that population and only 18 individuals could be found. So at that time, U.S. Fish and Wildlife Service made the historic management decision of managing for an endangered species by taking all wild individuals and bringing them into captivity. That had never been done as a conservation management endeavor before. I'm showing you a picture here of California condors because actually that year they made that historic decision twice. So not only were all black-footed ferrets taken out of the wild, but all California condors were taken out of the wild in order so that they would not go extinct in those very small remaining populations. So for black-footed ferrets, of those 18 individuals, they could only get 7 to breed. Black-footed ferrets and ferrets in general have a very complicated reproductive biology and breeding scheme. And so you can see here at the very first few years of captive breeding, they had a very difficult time getting them to breed, but eventually they did breed. In the early 90s, they had enough individuals that they actually were starting to reintroduce them back into the wild. And so in some aspects, this is a real success story, because if you look at the historic distribution of black-footed ferrets, we can see that they've been reintroduced across a vast portion of that historic range, all the way from Canada down into Mexico. But this isn't the whole story. Not all populations are equally successful. And let me just show you a little bit of these data. So this is a survey that takes into account the minimum number of individuals alive. So it's basically spotlight surveys. And then we have an axis here of year. And I'm just showing you 3 of the 17 reintroduced populations. And so for instance, let me show you the Wyoming population here. Reinforcement means that animals from captivity are reinforcing the wild population. So every year, animals from captivity are added to the wild population. For Wyoming, when reinforcement ended, the population declined and declined, but eventually it started to come back. In South Dakota, as soon as animals hit the ground, they started reproducing, and when reinforcement ended, they actually still considered to grow. So that population is considered self-sustaining. It no longer needs captive individuals. But then look at this Arizona population. Reinforcement occurs every year, but the population just continues to sputter along. And this is actually what most populations look like. Of the 17 reintroduced populations, only 2 are considered self-sustaining. And the reason for that, most people think, is because of sylvatic plague. So sylvatic plague is caused by the bacteria Yersinia pestis. And Yersinia pestis is not a bacteria that is native to North America. The sylvatic plague is native to Asia and Europe. It was brought in the early 1850s, and introduced into California, and it slowly crept eastward in rodent populations. It is 100% lethal to black-footed ferrets. The bite of one flea that contains Yersinia pestis has enough bacteria to kill that ferret, so they're incredibly susceptible. Interestingly, though, Siberian polecats and European polecats, which are the closest related species to black-footed ferrets, show some level of resistance to Yersinia pestis. So we know that there's a genetic component to resistance. So as Maud suggested, disease can be sort of the death nail to many endangered species. It may not be the reason why they became endangered, but when endangered species become critically small, any perturbance to that population can cause extinction. And so we worry about disease in things like Tasmanian devils with the infectious facial tumors, or certain bat species and white nose syndrome, or Hawaiian avifauna and avian malaria. And for black-footed ferrets, we worry about plague because it's really hindering recovery. And so with all of this talk of new biotechnologies available, biotechnologies, by the way, which are being used all the time in biomedicine, there's a lot of interest in using that biomedical technology in conservation, and in particular, gene editing to provide immunological resistance to pathogens of critically endangered species. So for black-footed ferrets, what would this look like? So this is a very simplified cartoon of immunological resistance. I am not an immunologist, and so I'm going to talk about this in the simplest of terms. But Mustela putorius, which is the domestic ferret, which is closely related to the European ferret, has these antibodies, which are immunological proteins that can actually detect bacteria. And in this case, the bacteria is Yersinia pestis, which causes sylvatic plague. These little baubles on the end of this immunological protein are very specific to Yersinia pestis, and they bind to the outside of that bacteria. When they bind to that outside of the bacteria, it allows the ferret to neutralize that bacteria. And so that is the basis for immunological resistance. For black-footed ferrets, they don't have this very specific bauble that can interact with Yersinia pestis to alert the body that there is this deadly bacteria in it. But what if you were able to change the genome of the ferret in such a way that it could then produce that protein? If that were the case, then the black-footed ferret might be able to resist that incursion by that bacteria. Now, this would have to be a heritable change in order for it to have usefulness on a population scale. So the next three slides are really three very simplified steps of how U.S. Fish and Wildlife Service is going about conducting experiments to see if this might be a feasible approach for black-footed ferrets. And so the first thing that they've done is to work on creating a successful mouse model of inherited antibody production. So in order to do that, you have to be able to clone individuals. That's the basis for creating any transgenic animal. So a transgenic animal is something that has a modified genome. So you create eggs, you modify the genome of those eggs by using what we call CRISPR-Cas9 technology. That's the gene editing technology that allows you to insert the gene. And in this case, the gene would code for that antibody protein into that egg. That egg grows up into a fetus, and then the produced pup is going to have inherited antibody resistance. So that's the idea. So that work is underway at Texas A&M University to see if these transgenic mice are actually resistant to plague, if the CRISPR-Cas9 technology works at inserting it. So there's lots of unknowns still within this transgenic mouse model. But step two, in order to get it into black-footed ferrets, you actually have to clone ferrets. And so this is something that's being done for conservation purposes as well, but it would also be needed to be done for making a transgenic ferret. For lots of reasons, including the fact that they have very complicated reproductive biology, as well as the fact that there just aren't that many black-footed ferrets, you need to use domestic ferret eggs, insert the black-footed ferret genome into that egg, create a blastocyte, which grows up into an embryo. You plant that into a surrogate domestic ferret, and if you're lucky, you get a cloned black-footed ferret. And so that is the basis for cloning a black-footed ferret. And that step has actually been successfully done. So a cloned black-footed ferret was produced in December 2020. That has not been able to be replicated since. So this is a very difficult process. And so becoming much more efficient at cloning black-footed ferrets is still a step that needs to be done. So this is not an efficient or highly successful process. And so that is still being undertaken by US Fish and Wildlife Service. But step 3, which is not happening yet because the first 2 steps are still underway, would then be to gene edit this black-footed ferret genome and continue this cloning process so that this resulting black-footed ferret might then have the heritable antibody response to Yersinia pestis. That is the overall goal of this gene editing process. So as Maud alluded to, these are really complicated processes that require a community of practice in order to accomplish. So in the case of the black-footed ferret, this involves for-profit companies, this involves not-for-profit companies, multiple academic institutions, and of course the US Fish and Wildlife Service, as well as the US Geological Survey, another federal research arm. So a very large group of people coming together to make this happen. Maud also alluded to the fact that there's a lot of ethical concerns about incorporating biotechnology into conservation. And that's something that we've worked very hard to keep in the process for including biotechnology in black-footed ferrets. So I was involved in a bioethical analysis of cloning of black-footed ferrets. And really this bioethical analysis is the study of this philosophical, social, legal issues and questions arising from incorporating this biotechnology into conservation. And it kind of just simply boils down to maybe you can do it, but should you do this technological advance, right? Is the technology well justified? Do you really need it? And is it being done responsibly so that animal welfare and ethics are being considered? Thank you very much.
Thanks, Sam, for the overview on what's going on with the black-footed ferret, and interesting to see where this field is going to develop. If there's any questions to Sam, you can put them in the chat or raise after the next talk. And with that, I'm directly handing over to Nikki from the University of Wageningen, who is giving a bit more detail also on what's following after gene editing. Nikki, please.
All right, yeah, thank you. And thanks to everyone for being here. There we go. Yeah, I'm going to talk a little bit more about this gene drive technology that Maud has talked about. And it's going to be a little bit more on the technical side, I'm afraid. And it's going to be about the evolutionary aspects of this technology. So biodiversity is in decline. And one of the main drivers are invasive species. And Maud has already talked about this, so I won't go into detail here. I will just jump right into the gene drive side of things. So Maud has mentioned if a certain individual is heterozygous for a certain trait, this is a normal inheritance scheme. It's inherited in about 50% of the offspring. What a gene drive does is it skews this inheritance ratio. So it would look something a little bit more like this, where if the gene drive is very efficient, it can be inherited in up to 100% of the offspring. So what this would look like in more of a population type diagram is here in black, you have all these wild type individuals. And then we introduce the gene drive in yellow. So the gene drive will skew its own inheritance ratio and start increasing in frequency. And correspondingly, the frequency of the wild type allele will go down. And if the gene drive is good, it will it will be completely fixed in the population. So how could we use this for conservation to control invasive species is by coupling the gene drive to a negative trait, as Maud has also mentioned. So then we'd start looking a little bit more like this, where the population size actually starts declining as the gene drive increases in frequency. And again, if the gene drive is good, if it does its job well, which is not a given, by the way, then the population will be completely suppressed. That would be sort of the goal with this technology. So to make that a little bit more visual, this is a project that I did during my master's internship. I did a model for the gray squirrel, which is invasive in the UK and other parts of Europe as well. And here you can see a spatial model. So in black are the wild type individuals, and then in yellow is the gene drive. You can see at the start they are kind of released in the middle here. You can see more and more yellow starting to appear, so the gene drive is reaching fixation, and then the number of individuals starts to decline. So this would be the ideal way in which a suppression gene drive would work. So what are the benefits of this technology? Maud has also talked about this already. It could be theoretically done in any species that is sexually reproducing, because the gene drive spreads over generations to the offspring by increasing in frequency. So that it must be a sexually reproducing organism. It is species specific because it spreads via matings. And it is potentially a more humane way to control invasive populations, because you could, for example, spread female infertility instead of, or as the negative trait that the gene drive spreads. It's also a very powerful, potentially a very powerful technology. And with that, of course, also comes the risk of safety. So these are some use cases that Maud has already mentioned as well. So in the gene drive research spheres, how did it all happen? So in 2013, this technology CRISPR was sort of discovered or developed. And then 1 year later, in 2014, CRISPR-based gene drives were already theorized. And people were like, this might be something that we could use. And then in years after, in several model organisms, gene drives were built. So fruit flies, yeast, and mosquitoes. And then in 2018, the first population suppression in a cage study, so a large cage of mosquitoes, was achieved. There were quite a few technical issues before that, but now in mosquitoes, it appears to be working quite well. Then last year, the first gene drive in mice that actually works fairly well was developed. So now it's also possible in more complex organisms than insects. And at some point, if we want to use this technology, which is a big question, of course, we're going to have to do field trials. And my project, so my PhD project, is actually about investigating a step that I think should be in between where we are now and field trials. So in my PhD, I'm looking into the evolutionary aspects of this technology, because, of course, in biodiversity, evolution is kind of the main force behind everything. And it could also be a force that is relevant for how well the gene drive works. So I'm looking both into the safety aspects and efficiency aspects. So let me just start with safety. Why do we need safety? That's kind of an open door to kick in, but anyway. So like I mentioned, the gray squirrel, for example, is invasive in the UK, and you can see here in this gray area, this is where the gray squirrel has completely replaced the native red squirrel. And then if we were to use a gene drive to help solve this problem, we would have to consider, of course, also the native range of the gray squirrel. If one gene drive squirrel were to escape or relocated to the US from the UK, that would be a huge problem. So we don't want that. Not only do we need safety, but we need inherent safety to the technology. So some of you might be familiar with this story about the rabbit virus that was used in Australia to combat the invasive rabbit there and to reduce population numbers. And at the same time, New Zealand decided that they didn't want to do this. They thought it wasn't for them. But nonetheless, rabbits with this virus were smuggled into New Zealand, which is one of the tightest, or a country with one of the most tightest biosecurity systems in place. So we don't just, we can't rely on this type of border control to make this technology safe. The technology needs to be safe in and of itself. One way to do that potentially is by targeting locally fixed alleles. So in order for me to explain that, I'll just take a quick step back and explain how a gene drive skews its own inheritance ratio. So you can see here in yellow, this is the gene drive, and this is made up of CRISPR components, which recognize this red sequence on the opposite strand, on the opposite chromosome at the same location. And there, because this sequence is recognized, CRISPR makes a cut, and oftentimes these are repaired by using the other chromosome as a template. So the cell is like, oh, my DNA is broken, I need to fix this. Let me just check the other chromosome and copy what's there, and then it's most likely to work again. So how can we use this specificity of the gene drive for safety? That would look something like this, where in our invasive population, we have this sequence that the gene drive recognizes completely in all individuals present, but then in a native population, it's not present at all. So what this would mean is if a gene drive was introduced into the invasive population, it would recognize the sequence and do the job. But if it were to escape to the native population, the part of the DNA that it's targeting is not there, so it can't work. And this would be inherent safety. Now, this is still a preliminary analysis that I'm working on, but most likely the situation is likely to look more like this, where the allele that you're wanting to target with the gene drive is not completely fixed in the invasive population, first of all, which is going to be a big problem for efficiency. And it is also present in a native population. So this is a precautionary, probably not on the locally fixed alleles as a safety mechanism. But there's still a lot of work to be done for this. So who knows? But there's a couple of evolutionary challenges that bring this about, that make it difficult for locally fixed alleles to exist. And that is, most invasive species have invaded their new territory fairly recently, in the last few hundred years. And that is really not enough time for evolution to have made these populations sufficiently different from each other. So they are still quite similar. And two other things are multiple invasions, which also exacerbate this problem, and large invasions. So when a lot of individuals get to a new place. And these are all problems that reduce the likelihood of locally fixed alleles to exist. So for inherent safety, it's probably not going to work. And we need to investigate other ways to do this, such as threshold-dependent drives and self-limiting drives, which stop spreading by themselves at some point, but I won't go into that. So that was the evolutionary aspect of safety, or the aspect of safety where evolution could help us. And then there is efficiency. So here's another open door. Why do we need efficiency? Well, if we're going to decide to use this technology, then we better make sure that it works beforehand. So this is an experiment done in fruit flies a few years ago, where you can see the gene drive frequency on the y-axis. So it starts at a low frequency when it's introduced, and then over the generations, it increases in frequency, like it should. But then it kind of stagnates at some point. So here, the gene drive is not powerful enough to actually work and to do its job. So why could this be? There's a lot of technical challenges that still remain, that a lot of people are working on, but there could also be an evolutionary side to this. So currently most gene drives are being tested in these lab strains of model organisms. So for example, in fruit flies, we have this white eye fly. So wild type fruit flies have red eyes actually, but this is a line of flies that has been kept in the lab for hundreds of generations, and it's very inbred and it's quite weak. It probably wouldn't survive if you put it back in nature. But this is what people are using for testing their gene drives, because it's of course the easiest organism. But when people have tested a gene drive in these white-eye flies, which is this bottom row here, it's about 62% efficient. Never mind what that means exactly. But then when they have tested the gene drive in more genetically diverse populations of fruit fly from all over the world, you can see that it already is much lower, this efficiency. So 55, even to 40. So this is a 10 to 20% reduction in efficiency in genetically diverse populations. And this is how that matters. Maybe you would think 10%, that doesn't really matter that much. But here you can see this model that shows the gene drive efficiency. If it would be 40%, then the population size would still decline and it would work. But then if you just reduce it by 5% or 10%, the story is already much different. So really small changes in efficiency are really important to investigate beforehand. So this is the second part of my PhD project, where I want to build upon this work. So I have a gene drive in fruit flies in the greenhouse here. It is a split drive for safety, no worries. And I'm going to test it in all these genetically diverse populations across Europe in the lab to further untangle what is going on here, because the gene drive is a very complex process, and there's still things going wrong that we don't know exactly why. And if we're going to use it in very complex populations, yeah, that's just going to be super difficult. So a lot of research still needs to be done here. So for my conclusion, we're going to definitely need good technical design with inherent safety mechanisms built into it. And locally affixed alleles are unlikely to provide that. And we need to look more into the evolutionary aspects of efficiency. And that is, of course, besides the fact that we need to also look into the ecological impact and public engagement, whether we even want to use this technology in the first place. And then if we do, regulation is also going to be super important. So there's still a lot of work to be done. But I think if we do decide to look into it, I think gene drives could be potentially a powerful new tool in our conservation toolbox. So I will end it there with my acknowledgements to my supervisors and everyone who's helping me. So thank you.
Thanks, Nikki, for this overview on gene drives. I think we got a really broad overview from the technical aspects on gene editing, cloning, and gene drives now, and the floor is open now for questions. So you might, I think you're all familiar with Zoom. So if there's any questions, just raise your hand and then you can speak, or you can also kind of write your question in the chat if there are any. And at the same time, Elena is going to share in a link in the chat as well. We have a very quick survey with the 3 questions. We ask you to kind of go quickly through while we do the discussions, so we know how to tailor potential future webinars. I've seen one question in the chat.
There are more questions.
Yeah. There was kind of Otto, did you want to kind of specify on the gene drive question or anything else? Nikki might want to check on.
Should I just read from the chat? Oh,
yes.
Sorry. Go ahead.
Shall I try?
Please go ahead.
Yeah, so possibly only my last remark. So if you call something, I can fully understand that it's important to have a clever design to maximize any, I would call it molecular confinement of a gene drive. But I would really never call it something like inherent safety. Yeah. This is really not, or possibly think of it whether you want to really use this in your communication. Yeah, because they're very theoretical concepts, they're not proven in the field. Yeah, and if you speak of evolution and of in terms of risk assessment, which is my field, yeah, it will be a real challenge to, for example, include rare events. What does a rare event mean, and what is the database you judge on? I already made a comment on one slide which shows how a gene drive would take over a natural population. Yeah, I think it is very important to understand that you cannot use deterministic models or very simple assumptions, like assumptions on a very simple mating complexity, yeah, which are not there in the field. So I think there's much more to do, yeah, and there are many other publications who will show that if you change the model assumptions, your whole spatial modeling will be changing. Yeah, so it is very case specific, and you need to be quite careful what to communicate. Yeah, but it is certainly not that simple.
Yeah.
Thank you.
Yeah, sure. Thanks for the note on the word of inherent. I will think about that. But yeah, you're for sure right that models are always simplified and that we need to do much more work there as well. Yeah, and I see your question in the chat, and you were talking about slide 4. And luckily that was not a model, that was just a diagram of the theory. So yeah, no worries, there's a lot of work.
Yeah, but you can make models like this, yeah, and models have been used like this, and it's I would always expect that more research is really being done of what are the critical model assumptions. You can also use a model and identify the parameters which may be important to know, and important to know from an ecologist perspective kind of before. Yeah? This is what I pasted in one of the publications from Verma et al., looking at mating complexity. And when you just take some examples which you know, like legs happen, yeah, mate choice happens, yeah, all these complex things, and you feed them into a model, and yes, the model then, what a surprise, shows you that legs do happen and mating complexity is quite high, and it has a very large effect on how genes are being dispersed, or inherited, and this is what I would want to have more research on that to make clear what you really need to do. And this would have, if you really want to put this into practice, then you would need good data and good argumentations why you do it in this way, and what kind of evidence you would need. And always, I think it is often forgotten that there's also a monitoring usually connected to it. And that can be very expensive, so that is also part of the complete package. If you want to see that option or opportunity, as you call it, it will be very important.
Yeah, I completely agree.
Thanks for that comment. I think there was a question on clarification, cloning versus gene editing or gene drive. I don't remember.
Gene
right?
drive.
Okay.
Gene drive.
Who wants to answer that quickly? Sam, Maud, is there anything?
Maybe.
Or is that I think I don't know, maybe the person who has been at a kind of asking the question, is there a difference between gene cloning and gene drive?
So when you try to clone an individual, it's just like you use the genome of an individual and you integrate it in another individual. I mean, in the case of the Blackfoot ferret, and gene drive is, so one is like really to provide, is not a strategy to to increase the variant in the population. Well, so the gene drive is really increase the inheritance of a modified or of a modification in a population. The cloning is more like you have a genome or a specific variant or individual, and you want to replicate that in the population. So the application is different, but that's true that's anyway. So is that clear? I mean, that's more or less the difference that I would see in cloning and gene drive. Gene drive really strategy to spread a different a variant in the population because you bias the inheritance of it. And then cloning is more like when you want to replicate a genome in a surrogate individual, or when you don't have that many individuals. Does that answer your question?
I think that goes back to the person who's been asking that. If not, please specify. Herr Otto nochmal?
Yeah, thank you. I think that comment shows something that what is very important also, that this definition. So what is genome editing? What is a true GMO? What is transgenic? Yeah, I mean the technique. So the methods kind of evolved. But I've written a comment on the American chestnut is no example for genome editing. It's a very basic old construct GMO, including antibiotic resistance marker. So maybe it will be of interest for you just to check what difference between a GMO which is regulated and some genome editing which may or may not be regulated would be. Yeah, certainly there is a qualitative difference what you change in the genome. Yeah? And this is important for you if you would change, what kind of changes do you want to make in the genome of a species? How would it affect it? So I would think that you would want to make as little changes in the genome as possible. So for example, you would only to include a resistance gene. But where does the resistance gene come from? What do you know where to insert it? What are the other genetic elements which are kind of involved if you insert it? So those would be also classical questions for the risk assessment. But I think it is also a kind of ethical thing. So if you speak of nature conservation and of conserving species and of protected species, we just had a viewpoint from our agency, and we found that it is legally not clear what it will be affecting protected species, whether you genome editing or whatever kind of do to it. And I think this is an ethical so a kind of aspect, but I would be very precise on the quality of genetic changes I would envisage for a species.
Yeah. Thanks for that comment. I think there's 2 points to it. I think a clear terminology is indeed important, and I think all speakers made it clear that there's kind of a lot, like it's been also kind of been in various documents. Okay, there's a lot of ethical and legal aspects to that. What we want to do, kind of give a brief overview on the uncertainty what these tools can do and they probably can't do, and where we are at that stage. So we don't want really to start the legal ethical discussion in that really short frame. I think it's absolutely important to do so, but this would probably need also additional speakers to do. So if there is a need to do so, then please add that in the questionnaire, and then we can focus on that. I also want to mention, and that would be really also interesting for many of you, that obviously this is also very different on different national levels, how that is seen and regulated. And Germany is probably one case, and others treated it differently. So that's also kind of a different story. If there's, are there any other questions and or comments at that stage? Then I would probably ask one to the speakers. We have been thinking quite a bit on the technologies and the scheme. Do you see within, we've been talking about the ferret, and we've been talking about the mosquitoes, and invasive species and rats. We do have, if I see that correctly, a mainly European dominated audience, also due to the time slot, obviously. To the three speakers, is there any species you would think kind of a gene editing and or a gene drive would be happening in the next 10, 15 years? And which kind of organism would that potentially be? I know this is a very tricky question. I've got that quite every now and then, and I just, is there anything you want to comment on?
So I can comment on a few efforts that I know of that are happening. So the Hawaiian avifauna and resistance to avian malaria, that's definitely actively being worked on. The resistance of coral to increased temperatures is another system that people are interested in using gene editing. Those are two examples that I can think of off the top of my head.
Well, in mammals and regarding the case of rats, probably the mice is going to happen before, which is also an invasive in some places. So you may have a better, I mean, the trials are first made in mice, and also because just mice are so well known in the labs as well. So I think that would be, and also, well, there is the case of the Lyme disease study in the US, and so that may be pushing the importance of the experimentations in that direction too. So which is not just the mice. But yeah, I think in Australia they also have a few things going on with mice. So on that side, I don't know about insects. Of course, mosquitoes are around, but yeah, the mammal side could be. Oh, I mean, yeah. So it's, but yeah, that's what I would say. Mice. Okay,
thanks, Maud. Nikki, would you kind of want to comment on the gray squirrel and how much that is reality, or is what's your kind of proposal? When is this going to happen, if at all?
Yeah. So first of all, there's still a lot of technical challenges because we just got gene drives to work in mice last year, and that is a super well-studied model organism. But then from the other side of things, I don't see a gene drive being used, definitely not within the first or in the next 10 years, because of all the work that still needs to be done with public engagement and figuring out if we really want to do this. Because with the gray squirrels, it's not just their impact on the red squirrels, which is native, but it's also deforestation and loads of other human causes. So I think we should really think about it really, really well, and that is on top of the technical challenges. So I don't know. I'm not entirely sure yet myself either.
Yeah. I think the main message probably to the whole audience is that none of these techniques is really ready to apply it immediately, and is under kind of development. There's one question in the chat from George Brett, and I think I passed that on to Nikki. From a modeling perspective, if you think about, I think this relates to gene drive, are better suited to R rather than K species.
I'm not entirely sure what that means, R selected species and K selected species.
I think it's R and K strategies. So George, do you want to specify if that do you want to, is that mostly on gene drives? You've been thinking. Yeah, okay. So it's gene drives. So our strategies would be in opposition to case strategies would be some with only a few offspring and others with many offspring and high fecundity. So I think mice and rats would be typical kind of organisms where you have lots of offspring. The question if they would be easier or it's more practicable than if you have like a long-lived species.
Yeah, right. So there has been modeling work that's been done on this subject. So you have species that live for multiple years and they have several reproductive seasons. And that sort of living, that life history slows down the gene drive by a lot. So the gene drive would definitely work better in the R-selected species then. Yeah.
Okay, good. Then we've clarified that. Herr Otto nochmal?
Yeah, thank you. I would possibly come back to the you named this viral example, and that's for me. So it is. I think it is much more easier to develop such an organism, than to think of all the background information and all the assessment concepts you would really need. So my suggestion would that if you really envisage something like this, you would need a good concept for how to put it into practice and what the risk assessment should be like and who should be responsible. And this is our concern, that this is not going to happen at the same pace as organisms are being kind of developed. So and I think that regulators will not be able to develop it because they will not have the funding for the research. So this need to be coming out of the scientific community as well. So it would be good if different universities, maybe who are not involved into the design of those organisms, would be concentrating also on the other aspects. Then even if you would, so I mean, think of standardization within regulation. It would need a test for pesticide testing. A new test battery, a new test organism needs around 15 to 20 years to develop. Just to give you an example, those are the timelines we are talking about. And this has been even when the whole assessment scheme and the whole concept is already there. Developing a single test species for non-target ecosystem testing takes 15 to 20 years. So this is another time scale we are talking about, or not many people are talking about. So I just want to mention this because this is from our side. This is what I see. Yes, developing a mosquito can be done within a year. But the ecological background information, how the population structure is, what this will mean for food web. So this is not there. And looking at pest management, you have so many pest species where the ecology is completely unclear. You just know that they feed on the crop. You don't know how they overwinter. You don't know much about it. Epigenetics. This will be scaling up if you modify wildlife. Thank you.
Yeah, I think it's important that researchers and kind of national or international and both bodies kind of work more closely together because it's going to be a long-term process. I want to hand over to Elena for some concluding or final remarks on the survey answers.
Yeah, thank you, Gernot. So we have 19 responses from the participant. As we would expect it, more than 70% are coming from research, but there are also people there are students and there are also practitioners with us, which is great. What do these 19 people think that the most following challenging topic in the future is going to be gene drive? And then the next one is the gene editing. And what you would like to in the future, for the future webinar, what you stress it's really important for you, it's even to how to use the genomic tool. So it means that you would like to hear more about genetic and genomic monitoring, even though any of training in conservation genomics, even metabolite coding, eDNA, and also ethical legal issues related to genetic technology and conservation. So there is a lot of things we can prepare the next webinar. Let's be in touch with us and we will keep you informed. And thank you for your participation.
Yeah,
so
thanks, Elena. That was good closing words. Thanks everyone for taking your time at this, at least in some parts of Europe, a hot afternoon. I wish you a nice evening, not too many thunderstorms. Stay safe, and we'll be in touch in one way or another. And next week we'll discuss on next webinars. Thank you all, and have a good day. Bye.
Thank you. Bye-bye.
Bye-bye.